Okay, good morning all. Good morning, good morning. Good morning everyone. Good morning. Okay, friends, please confirm me. Am I audible? Friends, please confirm. Am I audible? I hope I am audible, right? Perfect, perfect. Okay, so friends, in the yesterday class, we were discussing about the process of transcription. So while discussing the process of transcription, I mean to say, we have studied till the process of initiation. Okay, so friends, even though power go by any chance, if the power cut, please kindly don't leave the class within 10 minutes. I'll make sure that we will get the power back. Okay, please don't leave the class. We have 2 hours of class today. We have to finish the prokaryotic transcription also, as well as we have to finish the eukaryotic transcription also without fail. Okay, so friends, in the yesterday class, if you all quickly recall, we were discussing about the process of initiation. Okay, so friends, focus. The process of initiation basically takes place in 3 steps. One, we called it as what, friends? Binding of RNA polymerase enzyme. Everyone listen for 15 minutes. Formation of a closed complex and conversion of a closed complex to the open complex. Friends, every one of you please try to understand the point. This is what enzyme, friends? This is the RNA polymerase enzyme, you can see that. And this is what factor, friends? And this is what exactly we can call it as the sigma factor. And I hope every one of you is able to see here there will be a minus 10 nucleotide sequence, and friends, there will be a minus 35 nucleotide sequence. I have very clearly told you RNA polymerase never comes to know where exactly the promoter is located on the double-stranded DNA. That's why, friends, RNA polymerase will take the help of what factor, friends? RNA polymerase enzyme will take the help of the sigma factor. With the help of the sigma factor, RNA polymerase enzyme will come and associated with the minus 35 nucleotide sequence also, and minus 10. Basically, it will attach us to the minus 35. Now, every student try to understand what I am saying, friends. Listen, friends, my hand is the RNA polymerase. My fingers, you are able to, sorry, imagine my finger structure. This entire area we can call RNA polymerase enzyme. This palm region, we can call it as the sigma factor. So, all of you understand how this is the DNA. How exactly RNA polymerase will attach us? See, first my palm is sigma factor. Sigma factor will go and associate like this. So friends, sigma factor will associate very tightly with the DNA. All of you has to listen my statements very carefully now. You know, RNA polymerase associated with the DNA very tightly. And friends, where is the initiation region? The cap region is the initiation region. Everyone understand what I am saying? Now the RNA polymerase enzyme, from the promoter, it has to come to the initiation site. Then only, friends, it start the process of transcription. Once again, all of you listen to me. This is the RNA polymerase enzyme. This area is the sigma factor. RNA polymerase enzyme, with the help of sigma factor, it will associate it very tightly with this particular promoter region. That is what exactly, friends, we call it as the closed complex formation. Friends, every one of you please try to understand very clearly, if this particular open complex, if it cannot converted, sorry, if this particular closed complex cannot converted into open complex, then the process of initiation cannot occur. Anyway, look into this diagram. What is this open complex? All of you pay attention, friends. Look into the diagram carefully. This is the RNA polymerase enzyme. This is the sigma factor. All of you look into this. Now first, RNA polymerase enzyme, with the help of sigma factor, associated with the promoter, and it associated very tightly with the promoter. Suppose all of you listen my statements very carefully. Suppose this is the initiation site. From here onwards, the RNA polymerase enzyme has to start the process of the transcription. Every one of you try to understand, friends. If this particular RNA polymerase enzyme has to come to this particular initiator region, first of all, friends, the closed complex has to be converted into open complex. Now the question comes here. You can ask me, sir, how does the closed complex is basically converted into open complex? Now everyone will ask that, sir, if the closed complex is going to be converted into open complex, do you think is there any requirement of energy? How closed complex is converting into open complex without energy? I can give the answer to all of you that closed complex can be converted into the open complex just by the conformational change. Just by the structural change. There will be no requirement of energy. All of you listen. By the spontaneous structural change, by the spontaneous conformational change, the closed complex will converted into the open complex. Okay, friends, closed complex converted into open complex, and the RNA polymerase enzyme is ready now to start the process of initiation. Friends, this point is really important point. Just focus, everyone. Imagine, friends, this is the initiation site. Everyone pay attention. This is the INR. What is INR? I told you, initiator region, where the process of transcription will start. Now understand what exactly I am saying here. Whenever RNA polymerase enzyme reaches this area, which area? Whenever it reaches the INR, then only it start the process of transcription. All of you listen to me. Without reaching the INR, RNA polymerase enzyme should not start the process of transcription. But you know friends, everyone listen, RNA polymerase enzyme having really curious behavior. Because of the curious behavior of the RNA polymerase enzyme, you know friends what it will do. Before, listen, because of its curious behavior, before reaching the INR region only, it start the process of transcription. Now the question is very clear. Sir, do you think it will start the process of transcription very efficiently? Never ever. You know friends, it will start synthesizing the RNAs in the small pieces. And friends, this kind of a initiation we can call the abortive initiation. What do you call, my friends? Abortive initiation. Now you can ask me, sir, do you think this abortive initiation will be continued? Not at all. Once the sigma factor dissociated, RNA polymerase enzyme reaches this particular INR, then my friends, you all can see the picture very clearly. The process of RNA synthesis will occur continuously without interruption. So friends, I am summarizing once again. All of you pay attention. This is the RNA polymerase enzyme. This is the sigma factor. First, RNA polymerase enzyme associated with the promoter region with the help of sigma factor. Initially, RNA polymerase with the help of sigma factor associated with the promoter very tightly, which we called it as the closed complex. Later, my friends, closed complex can be converted into open complex just by the structural changes, just by the conformational change between the enzyme and DNA. Later, what is the unique statement I want you people to remember here? RNA polymerase enzyme, before reaching the initiator region only, friends, it starts synthesizing the RNA. This kind of initiation only we can call the abortive initiation. Why this particular abortive initiation occur? Because of the curious behavior of the RNA polymerase enzyme. Once the sigma factor dissociated, RNA polymerase reaches the INR. Friends, the process of transcription will occur very effectively. Kindly do not forget the statement. That is what, friends, the process of initiation. I want every student has to remember one more important statement whatever we studied. What is that particular statement, my friends? I hope every one of you remembered. In case of a prokaryotic organism, I said very clearly, transcription and translation are highly coupled in nature. Once again, understand, transcription and translation are coupled in nature. What do you mean by that? Which means, everyone understand, friends, when see here, every one of you look into this. This is the RNA polymerase enzyme. This is non-template strand. This is the template strand. My friends, which is coming out? You all can see, friends, RNA is coming out. Now here is the interesting point. Whenever friends, everyone understand, whenever 20 to 30 nucleotides of the mRNA comes out, that is sufficient. Immediately ribosome complex will come and attaches. Small ribosome and large ribosome will come and attaches. My friends, they immediately start the synthesis of protein. What, friends? Whenever 20 nucleotides of mRNA comes out, immediately ribosomes will come and attaches. They start the synthesis of protein. That's why transcription and translation occur simultaneously. What are, friends? Transcription and translation are highly coupled with each other. Kindly don't forget the statement. Okay. Now friends, how long the RNA is getting synthesized from the DNA until the RNA polymerase enzyme reaches a particular site. What do you call it, that particular site, my friends? We call it that particular site as the termination site. What do you call it, my friends? We will be calling this particular site as the termination site. Now friends, let's try to understand how does the termination occur. Termination occur in a two ways. One we called Rho-independent termination and Rho-dependent termination. So whatever the exam was held on the February 7th 2022, we got exactly a part B question from this particular termination process. So I am requesting every one of you friends understand the concept in a very clear way. Okay, just listen. Listen to me for 5 minutes very clearly. In the process of termination, there are two types are there. One is, my friends, listen to me, RNA on its own, it come out. It did not require any protein help. It did not require any enzyme help. RNA will come out of the DNA on its own. So just how with the help of I will tell you, just listen. With the help of a hairpin loop like structure, friends, it will associate with each other and it immediately come out. So it is not taking anyone help, my friends. That is the reason, friends, we will be calling this as intrinsic termination. My friends, instead of intrinsic termination, even we can use the word spontaneous termination. Anything is absolutely same. But sometimes it is a very difficult for the RNA to come out on its own. It cannot come on its own. So during that time, my friends, it will use certain extra proteins. What do you call it, that particular protein, friends? Rho protein. So if the process of termination occur with the help of a Rho protein, what is the statement that I want you people to remember here? We can call it as extrinsic termination. My friends, we can also call it as the non-spontaneous termination. Here the word non-spontaneous means the RNA never comes out on its own. That's why we are calling it as, friends, non-spontaneous termination. Now friends, let me explain to all of you how exactly this is happening. I'll show you the animation also. You will get a very clear idea. Friends, listen to me. This is the RNA polymerase enzyme. The blue color strands are the DNA and the black color one, what do we call it as, friends? RNA molecule. So every one of you listen, my friends. Whenever the RNA comes out, please listen my statement, friends. First 30 to 50 nucleotides of the RNA. Once again, everyone. First 30 to 50 nucleotides of RNA, my friends, it is rich of Z and C sequences. Once again, listen. First 30 to 50 nucleotides of the mRNA is rich of the G and C sequences. You all know very well G can pair with the C. Friends, whenever G pair with the C, all of you look into the picture carefully. It is going to form a hairpin loop like structure. You can see, right? This is the hairpin loop like structure. Friends, listen my statement. Now this hairpin loop, it start pulling the mRNA. All of you listen. Hairpin loop like structure start pulling the mRNA in such a way that because of its force, RNA which is associated with the DNA, it will come out. RNA polymerase enzyme also get dissociated with the help of that particular force. So RNA polymerase, hairpin loop itself pull the RNA without any external protein. Always remember, every one of you. Next, another very important point I want you people to remember here is that, friends, every single time, listen to me very carefully, friends, every single time this particular hairpin loop is not formed, even though hairpin loop is formed, friends, it is not at all stronger. It's not at all stronger. So I want you people to remember, during that time the RNA start using a protein called as a Rho protein. This is a very, very important protein. Now you can ask me, sir, how, listen every one of you friends, how does this particular Rho protein associated with the RNA molecule? That is the question. Friends, this particular Rho protein associated with a particular sequence present on the mRNA. You can see, right, on the mRNA there is a specialized sequence is present, which we called a RUT sequence. You know what is the full form of RUT? Anyway, you no need to remember, I am just writing. We can call it Rho utilization terminus. Rho utilization terminus. Now friends, via this particular RUT sequence, Rho protein will come and attach us. Then the Rho protein will provide the extra support for the RNA to pull it out. As a result, RNA can break down from the DNA and RNA polymerase enzymes get RNA polymerase enzyme get dissociated. Very, very simple point it is. I'll quickly summarize, friends. Focus everyone. Whomever did not understand, then later I will show you the animation. There are two type of terminations are there. One we called Rho-independent termination, or spontaneous termination, or intrinsic termination, where the RNA comes on its own. Next one we called, friends, Rho-dependent termination, which means, friends, it's a compulsory require the protein Rho. Compulsory require the external protein or an external enzyme called as Rho protein. Where exactly the Rho protein attaches? It will attach us to a specific sequence called as a RUT sequence. So that through that RUT sequence, Rho protein will come and attach us. Rho protein help the RNA to pull it out with highest accuracy and with a high speed majorly. By that, RNA can come easily. This is called as a Rho-dependent termination. So friends, now I am going to show you just 2 minutes of animation. In their role, in the animation only, I will show you how exactly closure complex will formed, how exactly open complex will formed, how exactly transcription and translation are coupled, how exactly Rho-dependent and independent termination will takes place. And I want every one of you to watch the animation correctly. Okay, look into it. Okay, so friends, every one of you please try to understand this particular concept very clearly here. This is what, friends, it's a bacterial cell. Inside the bacterial cell, you can normally understand that DNA. Now friends, please every one of you pay attention. You can see in the picture very clearly, friends, in this particular DNA, you can observe there are two type of promoters are there. You can see it very clearly. One promoter is located at -35 nucleotides, one promoter located at the -10. You can understand it clearly. Now friends, who is coming? You can see here RNA polymerase enzyme with the help of sigma factor it is coming. Now RNA polymerase and sigma factor together, what do you call it as, friends? RNA polymerase holo enzyme. Now look into this. RNA polymerase exactly come and attaches to the promoter region. My friends, everyone understand? Now this is the complex, we will be calling it as the closed complex. And friends, how does this particular closed complex is going to be converted into the open complex? And what did I tell to all of you? By the structural change or the spontaneous conformational change. We did not require any kind of the ATP at all. Now see here, you can see there, closure complex converted into open complex. Now see here, RNA polymerase enzyme will start synthesizing this particular RNA. See, in the picture it was not showing. Immediately once the promoter got cleared, sigma factor will dissociate. Now RNA polymerase enzyme reached a region. What is that region called as? INR region. Now all of you look into the diagram. Now RNA is getting synthesized. So friends, this is how the RNA will synthesize. Whenever RNA getting synthesized and when RNA comes out, all of you look into the diagram carefully. Now RNA is coming out. The moment RNA comes out, who will come and associate it immediately? Friends, immediately ribosome. You can see this picture, friends. Ribosome come and attaches. Immediately it starts synthesizing the protein. That's why, friends, in case of a see here everyone, protein is coming out. That's why in prokaryotes, transcription and translation are highly coupled. Now there are two type of terminations, Rho-independent, Rho-dependent. Every one of you look into the picture very carefully. In this particular Rho-independent termination, my friends, you can see this particular hairpin loop very clearly. What is happening with this particular hairpin loop? This particular hairpin loop pull the RNA on its own. Now see here, that's all. Now RNA came out. This is Rho-independent. But friends, when you look into the Rho-dependent, all of you look into this. See here, this is the Rho protein. Rho protein will come and attaches to this particular RNA. And it will pull the RNA very fast. As a result, what is going to happen, friends? RNA can comes out very easily. This is what do you call, friends? Extrinsic termination. Please kindly don't forget that. So that is about prokaryotic transcription process. That is initiation, elongation, termination. Friends, especially remember majorly what is sigma factor, how it is associating, what is closed complex, what is open complex, and how does the process of termination will takes place. Those points I want you people to remember. Okay, is it clear, friends, everyone? Now you can ask me any kind of a question from the prokaryotic transcription process. Now we will move into the eukaryotes, which is a very difficult process. See friends, I strongly believe when compared to the prokaryotic transcription, eukaryotic transcription is a very complicated process. The only reason behind that, friends, there is a involvement of many transcription factor, not one or two, my friends. There is a involvement of many transcription factors. That's good. I hope clear for everyone. Okay, now let's start with the transcription in the eukaryotes. Friends, I am requesting every one of you, seriously, this particular eukaryotic transcription is difficult. It is seriously difficult if you don't understand the concept clearly, friends. Sadguri, once the lesson is over, dear, once the unit 3 is over, once the unit 6 is over, in one day I will solve all the unit 3 questions from February 2022 paper. Same with this particular unit 6 also. In one day I'll finish the 2 chapters. Okay, don't worry, we don't miss any questions. Okay, so what I am saying, friends, this particular transcription in the eukaryotes is really a difficult process. If you don't understand, all of you listen, it will become more difficult, because the only reason is what, friends? There is a involvement of the many factors. and the process is not very simple process. So every one of you pay attention. And friends, all the time try to compare the transcription in the eukaryotes with the transcription in the prokaryotes. My friends, whenever you start comparing the transcription with the prokaryotes and eukaryotes, the concept is easy. Now I am requesting every student listen to me continuously for the period of 20 minutes without any interruption. Without any interruption, I want every student to listen this. Then automatically you will understand. Just pay attention and listen to me very clearly every single point. Friends, we studied very clearly in case of a prokaryotic organism, there is only one polymerase enzyme is involved. We all know very well. Now, in case of a eukaryotic organism, friends, there are 3 RNA polymerases are there. And I want you all people has to remember this. RNA polymerase 1, it is the one which is transcribing the rRNA in the nucleolus or nucleoli. RNA polymerase 2 is the major enzyme which is basically transcribing the mRNA. And friends, where does this particular mRNA is getting synthesized, friends? In the nucleoplasm. And friends, where does this particular RNA polymerase 3 and what it will do? All of you remember, RNA polymerase 3 is the one which is transcribing all the type of small RNAs. Very simple, friends. All the type of small RNAs can be transcribed by this particular RNA polymerase 3, like tRNA, 5S rRNA, all those, my friends. Okay, friends, every one of you listen my statements very carefully, friends. RNA polymerase 1, 2, 3, they are all start showing sensitivity with a fungal toxin named as alpha amanitin. Alpha amanitin is a powerful fungal toxin to which the RNA polymerases will show the sensitivity. There are many times, not many times, 3 times. The question was asked about this particular alpha amanitin. Friends, all of you pay attention. RNA polymerase 2 is the enzyme which will show high sensitivity. What do you mean by the word high sensitivity here? Here, friends, high sensitivity means RNA polymerase 2 can get easily inhibited by the alpha amanitin. When it comes to the RNA polymerase 3, friends, it is having a intermediate sensitivity. What, friends? It is having intermediate sensitivity. Alpha amanitin cannot show its action greatly on the RNA polymerase 3. Whenever, if you look into the RNA polymerase 1, friends, it is having low sensitivity. Overall, friends, it did not have the sensitivity only. So, friends, what kind of question that you can get in the exam here? You can get a question in the exam by saying that, listen my question, arrange the following RNA polymerases. They can ask either in the ascending order or descending order with their sensitivity towards their fungal toxin, that is alpha amanitin. So RNA polymerase 2 have the high sensitivity, 3 having intermediate, polymerase 1 having low or no sensitivity. So my friends, please pay attention, every one of you. If you quickly recall, we have studied the RNA polymerase enzyme cross section in the prokaryotes. No difference, friends, same here also. And I want every student has to remember one very important point. That is the core units. That is the core subunits. In case of a RNA polymerase enzyme, how many core subunits are there in prokaryotes? Alpha, alpha 2, beta, beta prime and omega. Friends, even in the core enzyme of the eukaryotic organism also alpha, alpha to beta, beta prime, omega. Core enzymes are same. Friends, in case of a eukaryotic organism, more than 5 subunits will be present. So I can say here 10 to 17 subunits are present. How many, my friends? 10 to 17 subunits are present. But what is the interesting statement? The interesting statement is whatever the core subunits that are present in the RNA polymerase of the prokaryotic organism, the same core subunits are present in the eukaryotic organisms also. Don't forget the point. In addition to this, little more extra enzymes are also present. Now friends, this is the DNA polymerase enzyme. Now friends, all of you look into the picture. This is what do you call as a downstream channel. We all commonly know that, we read it friends. So through the downstream channel, our DNA enters. We all know very well, RNA red color one, it coming out in 5' to 3' direction. We all commonly know, right? How do we add the nucleotides? We add the nucleotides at the 3' hydroxyl group. So there is a channel through which nucleotides can associated with each other. Don't forget it. Now friends, from this particular slide, I am requesting every student, please connect every single word that I speak. Seriously, if you don't understand this, it is really difficult. I will do my best of the best to make it extremely simple, and I'll make sure that you will understand everything in a clear manner. Focus. Friends, in case of a prokaryotic organism, what is the statement that we have understood? There are just 2 promoters are there, correct na? One is at the -10 and another one is at the -35. That's all we studied. Only just 2 promoters are there. But my friends, if you look into the eukaryotic organism, the promoters are very complex promoter. And the point is here, my friends, you should not think that there is only 1 or 2 promoters are present. My friends, many promoters are present. That all the promoters which can code for RNA polymerase 2. Why we are calling it as the class 2 promoters? Because they are all coded by the RNA polymerase 2. They can code for the RNA polymerase 2. There are 2 promoters are there, core promoter, proximal promoter. Friends, all of you listen my statement. If this is the INR, in the prokaryotic region where the promoters are present upstream. -10 and -35 extremely present only upstream. But here I want every one of you remember, in prokaryotes there are 2 promoters are there, core promoter and proximal. Friends, proximal promoters are only located at upstream. Upstream means left-handed or left side from the initiation site. Whereas friends, where does this particular core promoter is present? Core promoters are present even the upstream also, my friends. Downstream also. Friends, nearly 2 to 3 times the question was asked. What is the question was asked in the exam? The question says that core promoters are present both upstream and downstream, whereas proximal promoters are restricted only to the upstream, which is absolutely true point. Do not forget that. So my friends, first I will be explaining. Please listen to me carefully, friends. Don't miss any point. The concept will be 100% clear to you. There are first let's understand what are the types of core promoters, where they are located, and what are the type of proximal promoters. All of you look into the diagram very carefully here. Okay, this particular core promoters are, anyway I will draw here. I think I am covering the picture, you can't see that. I am drawing here, all of you look into this. This is called as INR region. Okay, so after that, all of you look into this, friends. After that, there are three promoters are there, which I am drawing in the yellow color. So you know what do you call it, that three promoters which are there in the yellow color? We called it DCE, downstream core element. So later, whichever I am drawing with the blue color, this promoter we can call it MTE, motif 10 element. I am drawing with another blue color, that promoter we called, friends, DPE, downstream promoter element. So later, apart from this, what else is present, friends? There is another promoter we can call Tata promoter, which is located at the upstream, and another promoter, my friends, we will be calling it as the BRE. One is called BRE, friends, another one we call it as the Tata. You can ask me, sir, is there any necessity that I have to remember all this? Yes, it is compulsory. You have to remember all these, where the promoters are located actually. It's very, very important, friends. So all of you listen. From the INR, what is this INR? Initiation region, where the process of transcription will start actually. Now friends, where this particular Tata box is located? From the INR, nearly, friends, minus 22 minus 26 nucleotides region. In between this particular Tata box is located. Friends, when you look into this particular BRE, from the initiator region minus 32 minus 46 region, this particular BRB will be associated. What is this BRE, friends? We can call the transcription factor to be recognition element. I will explain you in the upcoming slide. You will get a very clear cut idea. INR is located exactly at the +2 region. From the INR only you can compare what is the upstream region and what is the downstream region. And if you look into these three are completely what, friends? Downstream. We can call downstream promoter element, downstream core element, and friends, motif 10 element. Downstream promoter element will be located from +22 +32. Why I have representing plus, friends? Because it is located downstream. That's why I am representing the plus. When you look into the downstream core element, I have drawn in the yellow color, right? There are three downstream elements are there. So downstream elements also, friends, they are located from +18 to +52 nucleotide sequence. Motif element is located at +23 to +26 nucleotide sequence. Now the question is very clear. Sir, is there any necessity to remember the sequences also? I mean to say, where exactly it is located. Is there any necessity to remember their locations? Not required. What I want you people to remember for exam is that what are the examples of core promoters? That's all. And where they are, what are they? And what is the important statement you have to remember? Very important statement. That is, core promoters are located upstream also, friends. Core promoters are located downstream also. That is the major point I want you to remember. Now you can ask me a question. Sir, why do we require this many promoters? If you look into the prokaryotic organism, we read just two promoters. That's all. One promoter is located at -10 and -35. Sir, then why do we require this many? Because we are very complex organism. And yeah, listen everyone, the interesting statement. Along with the RNA polymerase enzyme, we are having many transcription factors. There are many number of transcription factors are there. Now friends, all those particular transcription factors has to come and associated with this particular promoters. So that's why we have many core promoters. And I also told you, right, core promoters are located where? Upstream also, friends, downstream also. But friends, if you look into the proximal promoters are located only upstream. Upstream means I can represent with what? Minus. See, at minus 40 to minus 70 nucleotide, there is a proximal promoter is present, which is rich of GC sequences. And friends, same minus 70 to minus 90 nucleotide, which means from the INR upstream only, there is another promoter is present, which we call cat box. These are all the list of core promoter. These are all the list of proximal promoter. Friends, before going to the process of the transcription correctly, I want every one of you to understand very clearly two things. I am requesting all of you pay utmost attention, friends. Then only you can understand very depth of the concept. Your concept will be very, very clear. Enhancer, I am giving you assurance. The question won't go beyond the level what we are discussing. So that's why pay utmost attention. Enhancer and silencer. Name itself indicating na enhancers are the one which can enhance the process of transcription. Silencers are the one which can silence the process of transcription. Very, very simple point it is. Now friends, every one of you listen to me. The interesting point here, look into this picture. This is what, friends, all of you understand. This is the INR. This is the INR, which means where the process of transcription will start. Friends, here there is an enhancer is located. This is an enhancer. Now my friends, you only tell me where is the INR and where is the enhancer. They are located very far from each other. Here comes an interesting point. What is that interesting point? Even though enhancers are located very far from each other, even though enhancer is located very far from the INR, still enhancer can enhance the process of transcription. There is no doubt about it. Same way, even though silencers are located very far from the process of very far from the INR region, still it is having the ability to silence the gene transcription. Don't forget that particular point. It is not necessary that enhancers and silencers are located very near to the INR region. It's not necessary. Even though they are located very far, they can do it. Now you can ask me, sir, how? All of you listen my statements in a simple way. Enhancers always enhance the process of transcription with the help of a transcription factors. Very simple. Enhancers, all of you look into this picture, friends. Every one of you pay attention. This is the RNA polymerase promoter, and from here INR, the transcription will start. You only look into this enhancer. See here, friends, this is the enhancer. You know why you call the enhancer is the distal control element. Why did I use a word distal control element? I called it as the distal control element because they are located very far. If they are located very, very close, then we can call proximal promoter because located very near. They located very far, that's why we can call distal control elements. My friends, this particular distal control element, they are associated with all these transcription factor and they will stimulate the process of transcription. So always remember, enhancer's function is what? Enhancing the process of transcription with the help of transcription factors. All of you pay attention, my friends. What is the function of a silencer? Silencer is the one which is exclusively involved in silencing the gene transcription. You know how it can silence the gene transcription? This particular silencers will convert the DNA into a condensed form or coiled form. So originally DNA is like this. But you know friends, what the silencer will do? Silencer will convert the DNA into a condensed structure like this. Now you only tell me, if the DNA get condensed like this, friends, everyone answer. Do you think, suppose this is the promoter, do you think RNA polymerase enzyme can recognize the promoter? Do you think transcription factors recognize the promoter? It is not at all possible. RNA polymerase feel very difficulty in recognizing the promoter. Transcription factors will feel very difficulty in recognizing the promoter. By that, my friends, if RNA polymerase is not associated with the promoter, how does the process of transcription will occur? Process of transcription never takes place. In a simple way, in one line, silencers basically block the process of transcription by converting the DNA into a most condensed form. Simple point it is. Then the next one, friends, enhancers are enhance the process of transcription by the help of transcription factors. Now, from now onwards, if you understand my lesson very clearly for 15 minutes of time, and I'll show you a very good animation also, if you understand this 15 minutes very clearly, the process of transcription in eukaryotes will be crystal clear to all of you. I'm requesting all of you friends, be patient and try to understand every single point, and I will repeat twice. By that you will get a clear-cut idea. There are two type of transcription factors are there. See, transcription factors are the one which are associated with the initiation, elongation as well as the termination, right? Now friends, before starting this particular process, I want to highlight one important point. This is an RNA polymerase enzyme. RNA polymerase enzyme having the C-terminal domain. This is really important statement, friends. It is the C-terminal domain. Every one of you listen. The C-terminal domain of the RNA polymerase enzyme having 7 amino acid. That's what we can call a heptad sequence. Once again, all of you listen. This is RNA polymerase 2 enzyme. RNA polymerase 2 enzyme having the C-terminal domain. This C-terminal domain of RNA polymerase enzyme having heptad sequence, means 7 amino acids are there. Why I am saying this 7 amino acid? Because all of you understand, very important friends, during the process of initiation, what should happen? This particular heptad sequence of the RNA polymerase enzyme will undergo minimal phosphorylation. What does it will undergo, friends? It will undergo the minimal phosphorylation. Friends, whenever the process of elongation is taking place, what will happen, friends? Maximum phosphorylation will take place. Maximum. My friends, whenever the process of termination is occurring, no phosphorylation or dephosphorylation. If you remember these 3 points, half of the concept is very clear. Minimal phosphorylation initiation, maximal phosphorylation elongation, no phosphorylation or dephosphorylation termination. Now friends, let's start. Don't interrupt me for 15 minutes. I'll explain very briefly. After that only you ask the doubts. Don't interrupt in the middle, please, because it's so confused. Now, what are the various type of transcription factors that are involved in the process of transcription? So transcription factors only will start initiation, elongation, termination. Everything is mediated by the transcription factors only. There are two type of transcription factors are there. Listen to me, I'll make it very clear. There are two type of transcription factors are there. One we called general transcription factor, promoter specific. Name itself indicating, right? General transcription factors. So they will be requiring all the time and they plays a key role in the basal level transcription. Promoter specific transcription factor. So those transcription factors will go and associated with the specific site of the promoter only. So that's why they will play a key role in the maximum level of transcription. Okay, so basal level transcription, maximum level. Anyway, so friends, first how I will explain to all of you is, I will explain twice what are the factors that are involved in the initiation. Later I will come to elongation, later I will come to termination, and I will show the diagrams at a time. By that the concept will become very clear to all of you. You people follow me very correctly and listen to me carefully. I will repeat twice. If you listen to me carefully, you will get an idea. Okay, there are many transcription factors are there in the eukaryotic transcription initiation. What are these? Transcription factor 2D. First of all, why we are calling transcription factor 2, friends? Because it is coding for the RNA polymerase 2, right? That's why, my friends, we called transcription factor 2. So transcription factor 2D, transcription factor 2A, transcription factor 2B, transcription factor 2F, transcription factor 2E, 2H. Total how many factors are there, my friends? Total 6 factors are there. Listen to me, friends, listen to me. This particular transcription factor 2D, friends, it is made up of, what does it made up of? It is made up of 2 components. What are the 2 components of the transcription factor 2D? One we called TAF, transcription activated factor. Another one we called TBP. I'll show you animation, you'll get an idea. TBP means Tata binding protein, and the TAF means transcription activated factor. So friends, every student please listen to me carefully. Where does this particular TBP will associate? TBP will go and associate with the Tata box. Listen to me, friends, please. Then TAF will go and associated with the INR. Very simple point. Transcription factor 2D contains 2 sub-factors. One we called TBP, another one we called TAF. TBP associated with the Tata box, TAF associated with the INR. Then what is the next factor? Transcription factor 2A. All of you listen statement, friends. Transcription factor 2A is responsible for to maintain, what to maintain? To maintain a stabilized interaction between TBP and transcription factor 2B that we are going to read now. What is the ultimate function of transcription factor 2A? Maintenance of a great stable interaction between the TBP and transcription factor 2B. And friends, what is the function of a transcription factor 2B? All of you remember 3 words. Yes, yes, yes. What is this yes, yes, yes? Transcription factor 2B plays a key role in the start site selection. Transcription factor 2B only will help the TAF also, where exactly this particular INR is located. So it plays a key role in the start site selection. So I'll quickly repeat, then we will move to the next factor. Listen to me carefully, friends. Transcription factor 2D contains 2 factors. One we called TBP TAF. TBP associated with the Tata box, TAF associated with the INR. You will see the diagram. Transcription factor 2A involved in maintaining the stable interaction between the TBP TF2B. TF2B involved in the start site selection. Friends, now I want every student has to remember very important statement about transcription factor 2F. You know what I can say? Transcription factor 2F is the most important transcription factor associated with the RNA polymerase from the initiation, elongation, termination throughout the RNA polymerase, or throughout the process of transcription. Transcription factor 2F always associated with the RNA polymerase enzyme. Every one of you listen my statements carefully. I can use a word. What is the word I can use? Transcription factor 2F having sigma-like activity. Transcription factor 2F having the sigma-like activity. Now all of you listen. Transcription factor 2F only help the RNA polymerase enzyme to recognize the promoters very accurately, and it is present with the elongation also during the elongation and termination. You will see in the diagram. Now transcription factor 2E, you know what you can call it as? It is the major enzyme which is basically required to recruit the transcription factor 2H. Very specifically, transcription factor E involved in the recruitment of H. Friends, listen to me carefully. After the transcription factor 2F, the most important transcription factor is the H. What is the speciality of H? Why H is important? The reason, friends, H will perform many functions, not only one. H is involved in the kinase activity. H is having helicase activity. At the same time, friends, H is also having ATPase activity. I am requesting every student remember this. Transcription factor 2H having kinase activity, helicase activity, ATPase activity. And my friends, transcription 2H is the major factor which is involved in the nucleotide excision repair also. And even this is the question asked in the part B also. Which of the following is the transcription factor that is involved in the nucleotide excision repair mechanism? We will read it. That is transcription factor 2H. So for your better understanding, I am quickly recalling. Focus. Transcription factor 2D made up of, see what we are talking right now is the initiation. When I show the diagram, you will get a very good idea. Here I can tell you, friends, hardly 5% to remember, 95% to understand. Pay attention. Transcription factor 2D made up of TBP, TAF, TBP associated with data box, TAF with INR. Then transcription factor 2A, it is only required to maintain the stabilized interaction between TBP and TF2B. TF2B is an important transcription factor which is required for the start site selection. Transcription factor 2F is the one which is having a sigma-like activity. It will be associated with the RNA polymerase enzyme throughout the transcription and act like a sigma factor. And transcription factor 2E majorly involved in the recruitment of 2H. 2H having kinase, helicase, ATPase activity. Whatever I have explained to all of you, I am same thing I am showing to you in the form of a diagram. Then you will get a crystal clear idea. All of you look into this, my friends. Let's talk about one by one. So first factor is what, friends? Transcription factor 2D. All of you look into this. It contains the TBP, it contains the TAF. So where this particular TBP associated, I already told you, it associated with the Tata box. Where does the TAF associated? Start point. After 2D, who will come? You can see, my friends, 2A will come. After 2A, who will come, friends? 2B will come. My friends, now I want every one of you has to understand the point very clearly. You can see here, this is the RNA polymerase enzyme. To this, what is associated? 2F. And what did I say to you? 2F is associated with the RNA polymerase enzyme throughout the process, throughout the transcription, it will associate. After the 2F and RNA polymerase enzyme, who will come? Transcription factor 2E will come. And what did I tell to all of you? Here comes the most unique statement requesting all of you to understand this. 2E is bringing what, friends? H. Look into this, friends. You can see now the red color one. That is the RNA polymerase enzyme. All of you please look into the point. RNA polymerase enzyme having the C-terminal domain. And what did I tell to all of you? If the process of initiation wants to occur, what kind of phosphorylation has to take place, my friends? Minimal phosphorylation has to take place. What? minimal phosphorylation has to take place. So how does this particular minimal phosphorylation will take place? Of course, with the help of transcription factor 2H. I told you it is having helicase activity, kinase activity and ATPase activity. So my friends, you all people know that how many strands of the DNA we require, my friends? We require only one strand of the DNA. So that's why we have to separate the strands. Who is gonna do that? Transcription factor 2H, because it is having helicase activity. You can see, my friends, it is opening the double stranded DNA. All of you look into the C-terminal domain of RNA polymerase, every one of you. It got phosphorylated. How much it got phosphorylated? Minimal phosphorylation. That's all. Friends, this is the process of initiation. Very, very simple point. You can see, my friends, initiation is started. Then entire transcription, right now whatever I am going to say, that is the most important step. Very important step now. This is initiation. After the initiation, what has to start? The process of elongation has to start. But I want you to understand one very important statement. All of you pay attention, utmost attention what I am talking about. This is what DNA. All of you listen, my friends. This is the INR region. This is the RNA polymerase enzyme. RNA polymerase enzyme and transcription factor 2F is there. All of you pay attention. Just now till here I have, listen to me, till now I have explained that process of initiation is happening, no doubt. Now all of you understand RNA polymerase enzyme cross the initiation site, no doubt. RNA polymerase enzyme cross the initiation site, it is moving downstream. Friends, every student listen. From the initiation site, 20 to 40 nucleotide downstream. From the initiation site, 20 to 40 nucleotide downstream, there is a site is located. You know what do you call it, that particular site? Very important point. RNA polymerase passing site. What was that? RNA polymerase passing site. What do you mean by that? Which means whenever our RNA polymerase enzyme reaches this particular site, it stop immediately. It never move only. It never ever move. Why? Because the site is called passing site. Once again I am repeating, listen. Whenever RNA polymerase enzyme start the process of initiation, from the initiator region, when RNA polymerase enzyme coming down 20 to 40 nucleotides, there is an important site is located. We called RNA polymerase passing site. At that particular site, RNA polymerase enzyme never ever move. Now the point is very clear. If the RNA polymerase enzyme is not moving, sir, how does the process of transcription will be continued? How does the elongation will be continued? In this particular condition, elongation factors comes into the picture. What are those elongation factors? I want you people to always remember short names. DSIF, NELF, the most important one, P-TEFb, positive transcription elongation factor B. I will be explaining to all of you, listen. At this particular passing site, 3 elongation factors will come immediately. What is the first one? DSIF will come, NELF will come. I always encourage you to remember the short names. Just listen. You know, friends, what these two will do? All of you listen. These two will help the RNA polymerase enzyme to become stable in this particular passing site. Because friends, now the RNA polymerase under heavy stress condition. Listen to me carefully. Now the RNA polymerase enzyme under heavy stress condition, because all of a sudden it got stopped. So this DSIF and NELF, you know what it will do? It will help the RNA polymerase enzyme to become stable. And here comes the most unique statement. Now these two are recruiting an important factor, positive transcription elongation factor B. This is the one which will do everything. What does it is made up of? Positive transcription elongation factor B made up of CDK9 and cyclin 1. What does it made up of? CDK9 and cyclin 1. Now you can ask me, sir, what is this particular P-TEFb will do? I have given a wonderful hint in the starting. If the process of elongation wants to occur, what did I tell to all of you? Maximum phosphorylation has to occur at the C-terminal domain. My friends, now you can see here, maximum phosphorylation occur. In the initiation, you can see, friends, minimum phosphorylation. Now all the amino acid present at the C-terminal domain get phosphorylated. Whenever it get phosphorylated with the help of this positive transcription elongation factor B, now RNA polymerase enzyme come out from the passing site and it starts moving very fast. Very simple concept it is. I know it's difficult. I am repeating for all of you. Pay attention. It's easy, friends. Follow me very correctly. You will see the animation. You will get a wonderful idea. RNA polymerase enzyme, whenever it crosses the initiation site, whenever it reach 20 to 40 nucleotide downstream, there is a site is located, we called RNA polymerase enzyme passing site. At that particular location, RNA polymerase enzyme will stop, can't move further. That time RNA polymerase is under high tension because it stopped all of a sudden. Then what is happening? There are two elongation factors named as DSIF and NELF. These two will help the RNA polymerase enzyme to become stable. At the same time, DSIF and NELF recruit the positive transcription elongation factor B. We can call P-TEFb. What it is composed of? It is composed of CDK9 and it is composed of cyclin 1. And these two together, what they will do? They will phosphorylate the RNA polymerase 2. Now RNA polymerase 2 completely phosphorylated, start the process of elongation. That's all. Very simple. Now I want every student has to remember one more very important point. Okay, friends, all of you look into the diagram, whatever I am drawing here. Okay, this is the RNA polymerase enzyme. This is how RNA is coming out, 5 prime, and this is the area where the nucleotides are adding. Friends, listen to me carefully. Whatever the nucleotides that are adding, how do we know whether we are adding healthy nucleotides or not? So there should be a proofreading. So who is doing the proofreading? That proofreading can be mediated by the transcription factor 2. Yes, it is the one which recognizes the nucleotide. If it identifies any wrong nucleotide is added, it will remove the wrong nucleotide, replace with the healthy nucleotide, transcription will be continued. Always remember, this is also asked part B question. Transcription factor 2, yes, involved in the proofreading. Whatever we studied so far, let's look in the form of a diagram. You will get a crystal clear idea now. And even though anyway, I am showing the animation also. All of you pay attention. Look into the picture, friends. This is the Tata box, INR. What is the first factor? Transcription factor 2D, followed by transcription factor 2A, followed by 2B, followed by transcription factor 2F, which is associated with RNA polymerase 2. Then transcription factor 2E, transcription factor 2H. What is the function of 2H? I said helicase, kinase, ATPase activity. So it is having a helicase activity. It open the double-stranded DNA. It is having the kinase activity. It will do the minimal phosphorylation. Very simple. During the process of elongation, P-TEFb, positive transcription factor elongation factor B, with the help of DSIF and NELF, what it is doing, you can see in the diagram. It will do the maximum phosphorylation. Upon maximum phosphorylation, the process of transcription will be continued. You don't believe the process of termination is extremely simple. I have given you once again hint. Remember the hint. Phosphorylation, Initiation require minimal phosphorylation. Elongation require maximum phosphorylation. Termination do not require any phosphorylation. When all the phosphate groups are removed out, automatically RNA polymerase comes down. Every student has to listen my statements. Whenever RNA, listen friends, whenever RNA polymerase enzyme reaches termination site, what will happen? Once it reaches the termination site, all of you look into this, transcription factor 2F will get dissociated. And but what is the point happening? Dephosphorylation occur. Dephosphorylation occur, at the same time transcription factor 2F also get dissociated. As a result, my friends, RNA polymerase 2 will become completely inactive. Inactive RNA polymerase enzyme comes down. This is the simple process of termination. Always remember, termination occur whenever there is a process of dephosphorylation occur. When the termination occur, whenever RNA polymerase enzyme reaches the termination site, then only dephosphorylation occur, then only RNA polymerase enzyme get dissociated. Friends, this is about eukaryotic transcription. Is that clear for all of you friends? You can ask me, anyone having any doubt, I will explain now. Please say yes or no friends. Is that clear for all of you? Please let me know.
Are you in class 12th and looking for a below 1000 rank in KCET exam? Then this video is for you. Myself will.
Okay, what about others? Come on, please let me know if anyone having any difficulty. I'll re-explain, please. Oh, that's great. So friends, I want every one of you to watch this particular animation. It will explain everything very elaboratively. Okay, whatever we discussed, you can see in the animation. So be patient and watch every point in the animation.
Hey everyone, quick biochemistry basics here. Let's talk about eukaryotic transcription. The enzyme required for the process of transcription is
watch every point very clearly, friends.
the RNA polymerase. The prokaryotes have only one RNA polymerase enzyme, while the eukaryotes have three RNA polymerase enzymes. RNA polymerase 2 is a major polymerase enzyme involved in the transcription of mRNA in eukaryotes. The RNA polymerase 2, along with other proteins known as the transcription factors, are required for the initiation of transcription. The transcription factors are also known as general transcription factors. Let's see how the transcription factors are designated. The initial two letters are written as TF, which stands for transcription factor. The 2 indicates that it's a transcription factor for RNA polymerase 2. The letter next to 2 can be A, B, D, E, F, and H, depending on the function of transcription factor. Because there are so many transcription factors involved in eukaryotic transcription, it's easy to memorize them with the help of mnemonics. For example, the mnemonics of TF2A.
I want every one of you to remember this particular mnemonics. A for apple, B for ball, D for dog, E for elephant, F for fan, H for helicopter, which are basically involved in what, friends? Initiation.
TF2B is ball, TF2D is dog, TF2E is elephant, TF2F is fan, TF2H is helicopter. Let's see function of each of them. In the first step of initiation of transcription, the transcription factor EF2D binds the Tata element in the promoter. The eukaryotic promoter is about 40 nucleotides long and located upstream and downstream of the transcription start site. About 30 nucleotides upstream to the start site, there is an AT-rich sequence known as the data box or the data element. TF2D has a protein called TBP which binds data sequence. TBP is also known as data binding protein. Once TBP binds data sequence, it bends the DNA by 80 degrees. This bending of DNA further helps in the binding of other transcription factors. This includes TF2A and TF2B. TF2A. TF2A helps in stabilizing the binding of TF2D with the promoter. TF2B interacts with TBP and the promoter region downstream to the data sequence. TF2B helps in the recruitment of RNA polymerase II on the promoter. Now, the RNA polymerase cannot bind the promoter on its own. A transcription factor TF2F helps RNA polymerase II to bind the promoter. TF2F interacts with TBP and TF2B while recruiting the RNA polymerase. TF2F also prevents RNA polymerase to contact DNA outside the promoter. Next, transcription factor TF2E binds the pre-initiation complex. TF2E helps in the binding of other transcription factor TF2H. TF2H is a very large complex with total 9 subunits. Out of 9, 2 subunits have ATPase activity. Using energy from ATP, it acts like a helicase and melts the promoter. And this finally causes transition from pre-initiation complex to open complex. The remaining 7 subunits of TF2H has a kinase activity. This kinase activity phosphorylates the C-terminal domain or the tail of RNA polymerase II, leading to promoter escape and transcription elongation. So we can remember the events of transcription initiation with the following mnemonics. The dog eats the apple and plays with the football. He gets tired and sits in front of the fan. When he sees an elephant, he runs away in the helicopter. Transcription elongation. Once RNA polymerase have initiated transcription, it shifts into elongation phase. The transcription factors that helps in elongation are called elongation factors. There are 2 such elongation factors, TFIIB and TF2S. TFIIB is recruited to polymerase by transcription activators. TFIIB is a kinase protein and phosphorylates serine residues in the C-terminal domain of the polymerase. This phosphorylation stimulates elongation. The other factor involved in elongation is TF2S. Now, the rate at which the RNA polymerase transcribes the DNA is not same at all DNA sequences. At some DNA sequences, the rate of transcription is fast, while at other DNA sequences, it can be slow. TF2S helps to increase the rate of transcription at the region where the rate of transcription becomes slow. It also does not allow RNA polymerase to pause and encourages to move on. 5' capping. As the RNA polymerase starts elongation, the mRNA starts forming. The formation of mRNA occurs in 5' to 3' direction. The first RNA processing event that occurs during elongation is the 5' capping. During this process, the terminal gamma phosphate of the nucleotide is removed by the enzyme RNA triphosphatase. In the next step, guanyl transferase nucleotide and etched guanine nucleotide. This on the resurrect SF stands for cleavable. Once the mRNA is cleared.
Okay. So friends, they are actually showing even the process of capping in the animation. That is not required. While we are reading this particular 5' RNA processing, I will explain briefly about the 5' capping, 3' polyadenylation and splicing. So in the animation, you have seen till the process of elongation. So after the elongation, what did I tell to all of you? When RNA polymerase enzyme reach the area of the termination, obviously it will undergo the process of dephosphorylation and the termination will occur. So friends, that ends the process of transcription completely in the prokaryotic organism also and eukaryotic organism also. All of you listen to me very carefully. Whatever the RNA right now we have synthesized in the eukaryotes, do you think it is a biologically active RNA? Not at all. It's not biologically active. It is an immature RNA. To acquire the biological activity, to make it functionally active, we have to do certain processes. What do you call that particular processes? So we can call the post-transcriptional processes. And where does this particular post-transcriptional process will occur? Obviously, they will be occurring in the nucleus, right? So how does this particular RNA processing or post-transcriptional processes will occur? We will read in the tomorrow class. I strongly believe everyone understand the concept of eukaryotic transcription very easily without any confusion, I believe. Okay, I really thank to all of you for your great patience. Okay, so I want you to improve your patience levels more and more. Okay, friends, that's all for now. In the tomorrow morning class, we will be doing the plant hormones. At least 2 to 3 hormones we are going to finish it in the tomorrow class itself. Okay. Okay friends, that's all for now. I will see you in the tomorrow class. Okay, thank you so much all of you. Bye bye. Thank you very much.